This is Lab 6, Enzymes. Just another reminder, your life is going to be easier if you do the lecture and all the reading and check off that box before you do this. That way you will understand the theory behind the experiment that we are doing. I'm going to upload a Word document, and this is a screenshot of the top half of that Word document. And so I thought I'd just go over it with you real quick. So, Enzyme Simulation Lab. Today, you will complete an online lab simulation simulating the effect of pH and substrate concentration on enzyme activity. So what that means is we're going to be looking at two independent variables. So that means that you must have two different experiments, and it's pretty simple to do that. The reason being that you can only have one independent variable at a time, like temperature, for example, and then you change it from cold to hot, and then you're going to look, see what's the effect on the dependent variable. All the other variables would need to be controlled. So for example, if temperature was your independent variable, then you would need to keep things like the pH and substrate concentration the same in all experimental groups. Okay, so I'll come back to that with a little more explanation later. The link is right here. Follow it. And this is in the Word document, so I heard a couple of you are like having to copy this down by eyesight. I'm sorry if that wasn't clear enough, but yeah, those should always be provided. And if they're not, absolutely post that one on the discussion board, and I'll get one up post haste, because I would not want to have to copy this myself. What a pain. So it's on the Word document. Okay, so modeling a good scientific protocol here. You start with a research question often, such as, how do substrate concentration and pH affect enzyme catalyzed reactions? Do they increase the rate? Do they decrease the rate? Or sometimes increase it and sometimes decrease it? Here are your directions. And once you have gone to the link above, so first go to there, right? And then you're going to read the directions on the left side. You do not need to do all of the things that they ask you to do. You simply need to know how the simulation works. So what you need to do is described below. So here is what you need to do, and of course, drum roll please, it's what you will be graded on. That's what we all are interested in. It wasn't that long ago I took some extra courses, so I know how you feel. You're like, just get to it, teacher. What am I going to be graded on? Okay, so number one, you're going to create two graphs during the lab, like the two described below. You can't see it yet. Remember to use appropriate units and intervals on the x and y axis to label each axis and to include a descriptive title. So title all graphs. Should be standalone. That's the idea of a good graph, meaning if I just looked at it, I'd be able to know what that graph is about. So I'm going to go to the next slide, and that just shows the bottom half of this Word document. Woo, there it is. So graph number one is going to be the effect of pH on amount of product formed per minute. That's I would say this is a so-so title, but I would make it a little bit more specific. Amount of product, such as what product? You could be more specific, I think. And actually, other than that, it would actually not make a bad title for this graph. So how to get the data? Remember, we are now, this one, pH is going to be the independent variable. So what that means is here, the amount of substrate added to each tube needs to be the same. When you graph your data, different pH values should be on the x-axis. Generally, the independent variable is always on the x-axis. There are some exceptions, though. And the amount of product formed per minute, units times 10 to the 6, that means million per minute, should be on the y-axis. So yeah, these enzymes are pretty fast, in the millions per minute. And then graph number 2 is a different thing. That's the effect of substrate concentration on the amount of product formed per minute. And I'm not going to describe that one. It's basically the same thing. Finally, when you're done creating those two graphs at that website, you're going to go to this fella right here, go to the animation quiz, or you can paste this link or this URL into your browser and go that way. Same thing, you know the drill. Watch the video, take the quiz, submit a picture, screenshot of your score. Minimum score is 80%, but remember, these are pretty good practice experiments for you, and I think that they will help prepare you for your next exam. Alrighty then, was there anything else? Yes, there were things. This is what you will see when you go to the first link. So this is where you're going to get the data to produce those two graphs. When you first see it, you won't see all this here. So you know what? Read the directions here, and they'll tell you how to do it. They'll tell you simple things like if you click on this computer, then it'll show you the graph, or not the graph, but the table where the computer would theoretically be recording it. You can click on this TV, and it'll show you a little animation. And I'm going to be nice and just tell you this stuff down here, you can just completely ignore it. I was fishing around with it. Some of them appear to be broken, actually. This is kind of an old link, I think. All right, this is what you are mostly going to be interested in here. So like some of our past labs, you can kind of push some buttons here. So right here, that's a pH button, and you can push the top one right here and it'll raise the pH. You can push the bottom one right here, it'll lower the pH. Same thing for this one, up, down. Right, great, that's pretty cool, pretty simple. I've included here one little sort of example of part of an experiment. What I did was, so I'm going to look at this, and then I'm also going to look at this here. So test tube number one, that's here, labeled number one. The amount of substrate added here, and remember they include grams, the units at the top or the heading of the column, as opposed to putting grams here, writing it all over and over and over again. That is bad form in science. So amount of substrate added, 0.5 grams. You added that. Well, actually I did. And where did I get it? I got it right here. See? So it says there's 0.5 grams of substrate here. And what you do is you just put the handy cursor thingy on it. You go grab, and then you put it into the test tube. And as soon as you put it into the test tube, this number right here will show up. It's a really fast experiment. And that is, the meaning of this is 72 somethings formed per minute. What somethings? Product. And it's in the millions. So that means that there was 72 million molecules of substrate formed per minute at the beginning of this reaction. Okay, now I'm going to erase some stuff. Huzzah! It's erased successfully. Are you all impressed? Well, if so, you're easily impressed. Okay. Next, you're going to be doing one experiment that looks at the effects of pH. So in that case, you're going to be changing these pHs to different pHs. Then you've got to add some substrate so that the reaction will occur. Now, here it's 0.5 grams of substrate. Here it's 1.0 grams of substrate, and it says that right here. I know the numbers are really small. But you need to make sure that you have the same amount of substrate to each test tube. Test tube 1, 2, 3, etc. Same amount, because pH is your independent variable here, and you want to know what the effect is on number of molecules formed at the beginning of the reaction. The other part of this experiment is that you're going to be keeping pH the same. Let's erase that. Okay. You're going to be keeping pH the same, and your independent variable is going to be substrate concentration. And so you're going to be adding different amounts of these substrates. There's 0.5, 1, I think that says 2 there, 4, and then 8 grams over there. Okay, where was I? Lost my train of thought. All right, so in that case, you're going to keep the pH the same. Now, what pH should it be kept at? Well, I'm going to let you decide, but it should be the same in all experimental treatments. One experimental treatment being test tube 1, a second being test tube 2, and then 3, 4, and 5. Ha ha. Okay, that is all for this slide. Next one. Ooh, it's blank. I guess I'll write something here then. So, how do we make graphs? So your results are going to be put in a graph like so, something like that. I'm going to give you an example where the independent variable is temperature, and the dependent variable is the same thing, amount, product, out of space per minute, you know, and then times 10 to the 6th. Okay, so When we are designing an experiment, we need to consider what intervals we're going to use. Our independent variable will be in the x-axis. It is temperature, and it's in degrees Celsius. You got to write that if you were doing one in temperature. Then you need to choose intervals, and they need to be regular. So for example, if you said, I'm going to do one experiment, one trial of this experiment at 5 degrees, then I'm going to do one at 90 degrees, and then another one at 91 degrees. This is going to give us data something like this. And then in 91, we got no data for some reason. This is not useful, all right? That's just silliness. So what you need to do, I'm going to need to erase a little more of this terrible example. What you need to do is have regular intervals. So like 5, 10, 15, 20, 25, 30, 35, 40, 45, 50, 55, 60. Okay, hope you enjoyed hearing me say all those numbers. Now I'm going to say them again. 5, 10, 15, 20, 30, 40, 50, 60, 70, 80, 90. No, you know, it must have been a long day. That doesn't make any sense. I just started going by tens. 25, 30, 35, 40, 45, 50. Okay, you get the point. All right, so you're going to run your experiment at all of those temperatures. So you're doing a lot of repeats in this case, or a lot of different intervals, and then your rate of reactions on the y-axis, and that's going to be amount product formed per minute times 10 to the 6th. And so we'll say maybe 1 million, 2 million, 3, 4, 5. I'm not sure how many it's going to be. If I did my first experiment and it turned out to be 50 million, then I'd have to redo that and write smaller units. But I'm just going to say that it's going to be 1, 2, 3, 4, 5 million is the most amount of molecules made in the first minute. So then you get some data, hopefully, and let's say the first data point at 5 degrees is 1 million, then at 10 degrees maybe it's 1.5 million, then 15 degrees it's a little more, and then I'm going to go a little faster here because this is probably beginning to sort of drive you crazy. And right here, this is often the optimum of a lot of enzymes that we're interested in. Because a lot of all enzymes come from living things. You can't make an enzyme, or at least we can't make one yet. And so often, because a lot of living things, well, mammals are the big one, insects, reptiles, and plants, their optimum temperature for enzymes is wildly different. Much hotter often, like plants can survive really high temperatures. But if it was an enzyme isolated from a mammal, then around 40 degrees, because that's above their normal temperature, which is, like if you're a human, 37. Above 40, it's going to start going down, and that is because the enzymes are denaturing. Probably will have nothing above maybe 50 or something like that. The enzyme is totally denatured. So then we've got a graph. Great. And you might be asking yourself, should I connect the dots? So that would mean just say like, all right, I'm going to do this like kid style and just connect them one by one like that, right? And then just draw a line between each one. And sometimes that is appropriate, and there isn't a hard and fast rule for when you should do that and when you shouldn't do that. The other option for drawing a line here is what's called a line of best fit. And that's what I'm going to do here, and I'll explain why in a moment. I'm going to start at the end and just kind of try to draw a curve that approximates where all those missing data points are. Okay, I tried to approximate where I thought all the missing data points would be. And what this would also help out with is if we had some really aberrant data. So for example, maybe this data point I just erased, maybe it was down here. So does that mean that you should, like in our first option, here's option 1, connect all the dots. All right, so, okay. All right, I'm going to connect them. Do you really think that that is an accurate representation of how the amount of product formed in the first minute of the reaction responds to temperature? I doubt it. Something funny happened here, and my suspicion is that's the real data. So you will often get what's called outlier data, like what I just showed you there. And a line of best fit is a much better way of dealing with that. So we're not going to do the childlike connect the dots. We're going to do that. And if you're writing this up, I'm not asking you to, so I guess you don't have to do this. And also, it's a computer simulation, so you're not going to get any aberrant data or outliers. But you wouldn't draw an arrow to it, but you'd say at 40 degrees, I think I added too little substrate. You'll try to make a guess as to what you think went wrong there. So you need to mention why it is that you drew a line of best fit that just ignored some of your data, because you cannot ignore your data. That's called bias, right? This looks like a really nice trend line here. It's like, oh, it goes up smoothly, then down smoothly, but then if you add in this like wonk, then it looks like you are not good at doing this experiment, and so you can't erase it no matter how tempting it is. Last thing on graphs. Should I connect this part here, that, to the origin? And the answer is generally no. Unless you can be confident that if you were to do an experiment for at 0 degrees, that's freezing, right? The water has frozen. Do you think any reactions are going to happen in ice? No, they're not. So if you can be reasonably confident that the true value at 0 for amount of substrate formed is in fact 0 substrate formed, then by all means connect the graph, as I have done there. Because I am very confident that if you're dealing with an ice cube and you put some enzymes and some substrate in the ice cube and it's frozen, there's not going to be any reactions happening. The molecules aren't moving fast enough. Okay, so that concludes the lab for enzymes, and you got to make 2 of these graphs. Read the directions that I'll post in the Word document, and it should be pretty clear. Feel free to post questions you have on the discussion board, or if you really want to, you can email them to me as well.